Good morning, and welcome to our explanation of cellular respiration. Okay, we begin with a big molecule of glucose, which is here. And the first process that it undergoes is the process of glycolysis. In the process of glycolysis, two phosphate molecules are added to either side, and two ATP are used to do this. So there's an energy investment phase, and then as it breaks apart, 2 NADH are produced and 4 ATP are produced.
Okay, so when these glycolyses are through glycolysis, this glucose is broken down into two pyruvate molecules, as seen here. Now, these pyruvates cannot enter inside the mitochondria themselves because they're too big to fuse to the membrane. So they have to, with the help of these transport proteins, they go through active transport to get inside the mitochondria. Once inside, they have NADH. They create NADH by reducing NAD+. CO2 is removed, and coenzyme A is added to create acetyl-CoA, which will be our main molecule in the Krebs cycle.
Okay, you can do the electron transport. Okay, so once we have acetyl-CoA, it goes into the citric acid cycle, and it undergoes around 10 steps, right? And then 2 CO2 are produced per acetyl-CoA. 3 NADH are produced. 1 ATP is produced, 1 FADH2 is produced, and as an end product we have oxaloacetate, which then can be re-put into the cycle, so it can continuously make more NADH and ATP and FADH2. And because this happens twice per glucose molecule, because it's split into 2 pyruvate, which is then made into 2 acetyl-CoA, the number that's produced is 4 CO2, 6 NADH, 2 ATP, and 2 FADH2.
Okay, so the 2 steps we've seen so far, glucose and the citric acid cycle, only produce a relatively small amount of the ATP in this whole picture scene. The part that creates the most ATP is actually this electron transport chain and chemiosmosis by the ATP synthase. So what happens is the FADH2 and the NADH created from here and from here, and also some of these become oxidized into these proteins here, part of the electron transport chain, and get converted back to their original forms of NAD plus and FAD. So the hydrogen is removed from each of them and goes through this protein into the inner membrane, which creates a gradient of H pluses in here. Now this gradient is very important for the next step, chemiosmosis, because of this gradient, the ATP synthase, which is a protein, is able to work these H pluses through the gradient. The H pluses go through them into here, and they use that power of going against the gradient to power the oxidative phosphorylation of ATP to produce these ATP molecules. Now this step actually produces most of the ATP, around 32 out of the total. And also water is produced from this electron transport chain as shown here.
Thank you.
I still don't get how the water